Hello, everybody. Obviously, I got dropped again. Sorry. Can you all hear me?
Yeah, we can.
Okay. Thank you. Just a few more slides and then we'll call it. Talking about HIV. HIV is still a very deadly disease. There are treatments for it, but they're not terribly effective in most people. There are some individuals that have lived with it for a long period of time, basketball players and things like that. But it's not, there is no cure, there is no vaccine for it, in spite of lots of money being spent. Okay, it's called GRID, gay-related immune deficiency. And the history is it was first discovered in gay males in San Francisco. If you have any interest whatsoever in learning about the early history of HIV, I strongly encourage you to read a book entitled "And the Band Played On." I've not actually seen the movie, but I have read most of the book. I say most of it because the lab I worked in, somebody had a copy of the book in the lab, and when I was waiting for centrifuges and things like that, I would read different chapters of it. It's extremely well written. It's written by a journalist, a gay journalist, who lived in San Francisco, talking about gay bathhouses and a variety of other things. It's a very interesting and scary book, not unlike the times we're living in now. It's first discovered in the US in 1981. I was married in 1978. At the time, there was some hysteria about how the disease was transmitted. Going back, however, we realized that it was the disease, the syndrome was originally described in Norway in 1966. There are older examples. It's been with humans for a long time. The oldest one that I'm aware of anyway came from what is now the Democratic Republic of the Congo, 1959. In 1969, there was a 15-year-old African-American that had the virus. What they're doing is they're going back and looking at plasma or tissue samples from individuals from a long time ago. Not entirely sure why hospitals keep these samples, but they have turned out to be very beneficial in tracing things like HIV. Norwegian sailor, 1976. Genetic analysis suggests that the most common strain dates back to the 1930s. Well, I shouldn't say the most common. Many strains date back to the 1930s, but there's evidence to suggest that the most common strain, M, goes back to about 1910. As I said, there are two different HIVs, one of which is derived from a monkey. HIV SIM is derived from. That is referred to as HIV-2, and the other is HIV-1, which is the one that's derived from chimps, and that's the one we're most concerned about. People in Africa eat what is referred to as bushmeat. It's not exactly the same thing as the open markets that you can find in Asia, but it's the same sort of concept in that wild animals are being harvested and consumed. Individuals that eat bushmeat are often infected by SIV, the simian version, but it's generally very weak, and the immune system can usually throw it off, can defeat it. It's thought that it takes rapid transmission from person to person for it to mutate to a much more infectious version. This is a somewhat dated, but still reasonably accurate indication of the distribution of HIV infection. You can see it's most prevalent in Africa, where it originated, particularly in South Africa and adjacent countries. There's all kinds of myths about how to cure it, including having sex with a virgin female is supposed to cure it. Talk to people that were in the forensic science field in South Africa, And they said it just resulted in an explosion of rapes of young females in an attempt to try to cure the disease. And of course, it was actually not effective and probably infected the victim with HIV. This just gives you some illustration of the distribution. You can see that the most heavily infected countries, I think this is from 2014, are mostly found in Africa, although there's a fairly high incidence in India as well. A couple of links, YouTube videos that you might want to watch that are pretty interesting. As I said, the most interesting book I've ever read dealing with it is the gay scene, basically, in San Francisco in the early 80s, late 70s and early 80s. And the band played on. Very well written. The author eventually died of AIDS. There are different types. As I said, HIV-1 from chimps, HIV-2 came from monkeys. Their immunopathogenesis is not identical, although they can both end up in AIDS. HIV-2 is mostly a long-term infection. Its progression, probably shouldn't say non-progression, but its progression is not guaranteed. It doesn't progress in some individuals it does progress, but the progression is much slower. Some with HIV-2 do get AIDS, similar to HIV-1. HIV-2 is found primarily in West Africa and in Portugal. West Africa used to be colonies of Portugal, which is why there's a linkage between the two. HIV-1 is global, found in every continent, every country, except Antarctica. You can have a dual infection, referred to as HIV-D. There is some evidence, mostly anecdotal, that infection with HIV-2 may protect from infection by HIV-1. But again, it's not reliable. You certainly wouldn't want to risk your life going out and intentionally getting infected with HIV-2 in the hopes that it would provide protection against HIV-1. As I said, it's anecdotal as much as anything else. Some studies have supported it, some have not. Oops, got to delete that. That's an indicator of where I stopped previous year when I was teaching it. We're going to go a little bit past the normal hour just so there's some substance in this version of the YouTube video. Very fast replicating, 10 to the 9th, 10 to the 10th virons per day. That's huge. It's highly mutable. Mutation rate of 3 to 5 every 1,000 base pairs, basically. That's because the reverse transcriptase does not have a proofreading mechanism. And so you have a lot of mutations that occur whenever reverse transcriptase is involved in replicating a genome. The genome is relatively small, 10,000, and on average, there's 2 mutations per genome. Because of this high mutability, oftentimes the virus will outrun or develop immunity to drugs and the immune system. Can also recombine, which is a scary thing, although it turns out it wasn't as big a deal as we feared it would be, with other viruses, creating novel viruses. At one point, the concern was it was going to recombine with cold virus or a flu virus and become much more infectious. The reverse transcriptase has the ability to jump back and forth between different strands, creating hybrid molecules, basically. So there's a lot of opportunity for the genome to rapidly evolve. Treatments consist of nucleoside analog, that is molecules that mimic nucleosides, the nucleic ribosomal, the ribosome, ribonucleotides, and will inhibit reverse transcriptase. You also have non-nucleoside analog, meaning they don't mimic the nucleotides, reverse transcriptase inhibitors, and you also have protease inhibitors. Part of the maturation of the virus requires protease activity. So there's a three-prong attack in most cocktails to treat HIV. Yep, cocktail of the reverse transcriptase inhibitors as well as protease inhibitors. Unfortunately, only about 50% is this effective in terms of long-term treatment. In roughly 50% of patients, the virus develops resistance to this treatment within about 6 months. One more slide and then we'll go. CCR5 is another protein found on the surface of our cells. This is one of the receptors that fuses with CD4 at GP120 upon initial infection. Some people are mutant, however, for this co-receptor, CCR5. There's a deletion of this gene, delta 32, which provides at least partial protection from smallpox as well as the plague, Black Death, but increases sensitivity to the West Nile virus. As you might expect, populations that were most heavily exposed to smallpox and the plague, mainly Northern European, have the highest incidence of this delta 32 CCR5 allele. Provides, even in the heterozygous state, provides partial resistance to HI1 infection, at least the most prevalent strain, the M strain. It was discovered when a gay couple, one of the couple was infected with HIV and succumbed to AIDS. The other partner, they did not practice safe sex. The other partner was constantly exposed to HIV from the infected individual. That individual never became infected. And turned out that individual, the resistant one, was homozygous for CCR5 delta 32 allele. It's found in, it's estimated roughly 40 to 20% of Europeans are heterozygous for this particular mutation. Again, the original selection pressure was resistance to smallpox and Black Plague. And it just fortuitously turns out it provides protection from HIV infection as well. The highest frequency are in Nordic peoples, decreases as you move south. Again, that corresponds with particularly smallpox, but also Black Death. There's been speculation about the possibility of using RNAi to knock down CCR5 in individuals that don't carry the CCR5 allele with the delta 32 deficiency. And we'll just call it quits there. We're going to review briefly RNAi, RNA interference, RNA-dependent gene silencing. That's where we will pick up with next time. Does anyone have any questions? No? Okay. Just to remind you, I am recording these. Certainly by the end of the weekend, these should be available on YouTube, the recorded versions of it, the lecture, the slides and whatnot. I will be sending out an email telling everyone that they are available and how to access them, what to look for. And I will also, for the 6 of you who haven't gotten your exams yet, you will be receiving them hopefully by the end of today, tonight, sometime, if not, certainly by tomorrow. Okay, guys. That's it.